Hello, good afternoon everyone. My name is Mohd Nazri Ismail from University Science Malaysia, Penang. First of all, I would like to thank the organizer of the International Online Analytical Science Conference 2022 for giving me this opportunity to give a talk today. The title of my talk is the LC-MS-MS-based proteomics, advancing biotechnological and biomedical research. Today I will talk on two main projects. One is related to the biotechnology of rubber latex, and the other one is related to the biomedical aspects of the diabetes. Okay, on the first project, it is on the profiling of proteins and unrestricted identification of post-translational modifications in the Hevia brasiliensis latex. The rubber tree, which originates from the Amazon rainforest, is the primary source of natural rubber, which is applied in transportation, constructions, machineries, medical equipment, and etc. So, what motivates us to study the proteins and PTMs in plants? First, to date, there is no complete rubber tree proteome data available. Proteins and PTM believed to regulate the latex biosynthesis pathway. In addition to that, acetylation also known to involve in transcriptional regulation, apoptosis, and cytokine signaling. And finally, the phosphorylation regulates carbon and nitrogen metabolism, synthesize sucrose and ammonia, and involve in plant growth and response to stress. The problem statements. Even though rubber is a very important commodity, the biosynthesis pathways are still understudied. Furthermore, the species-based plant PTM database did not cover this tree. And lastly, even though there are many different tree clones, but their performance were never compared and correlated with the biological pathways. So in this research project, we aim to identify the proteins and PTMs of rubber latex and then to correlate the expressed proteins with the molecular weight distribution of latex from different rubber tree clones and ages. Now let's move on to the method. Following fresh latex collection, we separated the latex into three different parts, which is known as serum, lutoids, and rubber particles via ultracentrifugation. The fractionation will help in the protein identification. Each latex subcellular fractions are then undergone purifications, protein extraction, further fractionation, protein in solution digestions to digest proteins into peptides, and then straight to LC-MS-MS for protein and PTM identifications. In our methodology, we perform de novo sequencing and also in silico, followed by in silico validation for the PTMs. And these are the two main databases that we have used in our methodology. And this is the equipment that we've used in our laboratory, the liquid chromatography tandem mass spectrometer, which is the thermoscientific Orbitrap Veloz Pro hybrid ion trap Orbitrap mass spectrometer coupled with the Nano LC from Proxeon, which also this instrument contains the three different fragmentation techniques, which is the CID, ACD, and ETD. Following the analysis, the raw data from the LC-MS are then analyzed by using PeakStudio 8 for de novo sequencing and database matching, including PTM identification. At the same time, we also perform the molecular weight and molecular distribution analysis. Instead of using the fresh latex, we use dried rubber for this analysis. So briefly, the dried rubber is first dissolved in tetrahydrofuran and then gone through further analysis, further processing before analyzed by using the HPLC Agilent 1200 with a refractive index detector. Now let's look at the results. This is the protein identification results. So the databases listed here, the photosynthesis, latex allergens, rubber wood formation, latex biosynthesis, and disease resistance are the database that have been annotated from a previous work on the genome sequencing of the rubber tree. So we have matched our protein identifications that came from two different mass spectrometric methods that we term them as proteomics and glycoproteomics. So we matched with the expected proteins from these databases, and we can get a consider high percentage of protein identification. Other than that, we also matched the public database, the SwissProt, and also the plant GDP. Moving on to the PTM results, so phosphorylation and anti-acetylations are the two main PTMs that we have studied and focused in this project. So phosphorylations that are commonly found on the STY and the acetylation that are found on the N-terminal amino acids. So these are the number of proteins in these samples that were identified to contain these two PTMs. This is an example of the peptide and PTM sequencing that have been done on our LCMS data by using the Pig Studio software. So in this peptide, you can see this is the sequence of the peptide, and the small letter S, which is the serine, is the modified serine. And in this case, it contains this phosphorylation on that amino acid. Whereas for this peptide, is a peptide that contains the N-terminal acetylation at the alanine residue as shown here. Since that we have the capability to perform three different fragmentation techniques, so we have compared the performance of each fragmentation techniques in detecting phosphorylation and N-acetylation on the rubber latex proteins. So for phosphorylation, we found that CID, collision-induced dissociation, was better at detecting these phosphorylated peptides compared to the HCD and ETD. Whereas for the N-acetylation, CID, both CID and HCD, which is the high energy collision dissociation, work better compared to ETD in detecting acylated peptide. So we also identified that most of the phosphorylated proteins are those that can be classified as allergens. Whereas for the N-acetylated proteins, which include proteins that involve in latex biosynthesis, such as the small rubber particle proteins and the rubber elongation factors. So all are very important proteins in the rubber biosynthesis. So this graph summarizes the molecular weight distribution data according to different tree age group 5, 7, and 10. So you can see generally the younger trees have the highest molecular weight distribution index compared to the older trees. So this is still the same data, but this time we plotted according to the different clones, and you can see that each clones have different pattern in terms of the molecular distribution as the tree grows older. So the conclusions to date for this project is that the identification of the proteins that we have done has confirmed the existence and the mechanisms of the previously proposed pathways such as the photosynthesis, the latex allergenicity, the latex biosynthesis, rubber wood formation, and disease resistance. And there were 61 peptides from 45 proteins from H. brasiliensis protein sequences from UniProt KB that were identified and predicted to be phosphorylated or N-terminal acetylated. Most of the phosphorylated and N-terminal acetylated sites discovered has yet to be reported in UniProt KB. And this will provide great assistance in the research of the functional properties of H. brasiliensis latex proteins, as well as being useful biomarkers. As the molecular weight distribution data was actually very fresh, came out from the laboratory, so what we want to do next is to find the correlation between the rubber molecular weight or molecular distribution and protein and PTM profiles according to the tree age and the clones. Now I would like to move on to the next project, which is to explore the diabetic biomarkers using proteomics. The first sub-project looked at the potential urinary disease marker for diabetes. Diabetic nephropathy, or DN, is one of the renal complications that can happen in diabetic patients. The end may lead to end-stage renal disease eventually if the patient is left untreated. So in terms of markers, there are already markers for the end diagnosis. For example, albuminuria, but it's not a suitable one actually because it is lack of specificity and sensitivity. And there are actually other markers that have been proposed and identified as well for this disease. In kidney diseases, the structural and functional proximal tubular changes have been shown to affect the increased excretion of proteins with molecular weight less than 40 kilodaltons. So meaning that there is a higher increase of proteins less than 40 kilodaltons. And previously have been shown that this proteomics classifier CKD273, which is a panel of 273 differentially expressed urinary peptides, was elevated in CKD patients compared to healthy controls. And these peptides were found to be associated with prediction of progressive DN and CKD. So the objective of this work were to analyze the changes of low molecular weight proteins in urine of diabetic rats and pre-diabetic rats by the effect of diet, namely the normal diet and the high-fat diet, and also to evaluate the effect of metformin treatment to urinary protein profile. This is our experimental design. So in total, 60 male rats were used and 48 were assigned as the test group. So in the test group, the diabetes was induced in them prior to urine collection in the next morning. So this is the groups that we have created. So in the control groups, they are non-diabetic. So we have two different treatments, so normal diet and also the high-fat diet. And the experimental groups, we divided them also in the diabetic and pre-diabetic groups. And in the diet and treatment, we have normal diet plus metformin, and then also we have the high-fat diet followed by high-fat diet plus metformin, and this is the same for the pre-diabetic groups. Next, the urinary proteins were precipitated from the urine, and then the concentration were determined. This is followed by STS-page. So the reason we do STS-page first was to look at the existence of low molecular weight proteins, and then the selected protein bands were submitted for the LC-MS analysis before the data was analyzed by using the Pig Studio software. There were two bands that captured most of our interest, which is the band at the molecular weight 23.5 kilodalton and 11.9 kilodalton. For the 23.5 kilodalton band, diabetic rat showed a higher intensity compared to control and pre-diabetic, whereas for the 11.9 band, it was seen higher in the normal compared to the diabetic and pre-diabetic rats. Okay, there were also a number of lower intensity protein bands detected with different expressions in the different groups. So here we listed 7 protein bands, and if you can see the protein bands 15.9, 14.9 and 9.8 were consistently detected in healthy and pre-diabetic rats, whereas the other bands 27, 19.9, 16.2 and 9.4 were not detected in any of the healthy and pre-diabetic rats but show inconsistent excretion in diabetic rats. So because we want to focus on biomarker for early detection, so we have focused on the bands that are consistent in the pre-diabetic rats. So we have identified proteins 23.5 at the Ig kappa chain C region, and then protein 15.9 and 40.9, both are the major urinary protein. And lastly, the protein 9.8 kilodalton was the histone H4 protein. The 23.5 kilodalton protein bands was found consistently in the urine of all rat groups, but it was found in the higher intensity in the diabetic rats. So the same protein bands was consistently high in diabetic rat regardless of the diet or treatment given by metformin, and was found lower in healthy and pre-diabetic rats. So these proteins 23.5 kilodalton can be a good indication for diabetic condition which is not affected by the external factors such as diet and treatment. However, it's still we need to quantitate this protein for further analysis. The other bands like the 15.9, 14.9 and 9.8 were also detected consistent in all groups of healthy and pre-diabetic, but were found missing in the diabetic group. So these protein bands can also be a useful biomarker for diabetes. As shown in the previous slides, the 23.5 kilodalton protein is the Ig-kappa chain protein, and this protein was found to be unregulated in type 2 diabetes mellitus, and it excretes from early years of T2DM up to the cause of diabetic nephropathy. And screening of visceral adipose tissues of pre-obese diabetic subjects showed an increased abundance of this protein compared to the pre-obese subjects. Whereas for the 15.9 and 14.9, which was found to be the major urinary proteins MUP, and these proteins was found to be involved in glucose metabolism by suppressing hepatic gluconeogenesis. And in our research, diabetic rats also identified a down-regulated expression of MUPs. Finally, the 9.8 kilodalton protein, which was identified as the histone H4, and histone H4 is known to involve in the regulation of gene expression. So we know that the pathogenesis of diabetes is related to histone post-translational modifications, and it can affect genetic expression in organs linked to diabetic complications. So overall, from our protein profiles, comparison of these animal groups, we have shown that there are a few proteins that were found to be useful to indicate the condition of early diabetes, and the excretion of these protein markers were shown to be not affected by the diet intake and treatment. Now, I would like to bring your attention to our second sub-project, which is the identification of brain proteins in mice with high-fat diet. So dietary fat is one of the major factors contributing to energy intake related to obesity. The acceptable micronutrient distribution range for a total fat is 20 to 35%. So beyond this is already considered high-fat diet. The chronic high-fat diet consumption can result in weight gain or obesity, increased systemic inflammation, oxidative stress, and destruction of glucose homeostasis. Dietary fats also impact brain's processes by regulating the synaptic transmission, membrane fluidity, and signal transduction pathways. On to the methodology for this project. So in total of a total of 12 rats were used in this project, and then we divide them into two groups, one for the normal diet and the other one with the high fat diet. And then we harvest the animal, we obtain the brain tissues and ground them into fine powders. We also perform the measurement of blood biochemical parameters and key health factors. We perform 2D gel electrophoresis, which is then followed by in-gel digestion of proteins to release the protein from the gel into solution by using trypsin as the enzyme. Then the spots were then selected and submitted for LC-MS analysis for the identification using the same instrument with the previous project. And then we perform statistical analysis by using the student t-test and then two-way ANOVA. Let's move on to the results. This table summarizes the data that we obtained for both the normal diet rats and the HFD rats. So you can see first at the food intake, the rats for the normal diet has higher intake because they need more energy compared to the HFD. Whereas for the calories intake, also the HFD is still higher than the normal diet, but not that significant. And then the serum cholesterol almost similar for both, for the adipose tissue, and then the percentage of adipose tissue for body weight. Also, it's higher in the HFD compared to the normal. And then the efficiency, metabolic efficiency, normal rat has higher metabolic efficiency compared to the HFD. So this image showed the selected spots for the proteins from the rat with normal diet and high-fat diet. Overall, the number of protein spots detected are quite similar between both groups, which is about 145 spots. Two spots, the spot 6702 and spot 5201, were upregulated at significant levels in the high-fat diet compared to the normal diet. And these protein spots were identified as the heat shock cognate protein and the pyruvate dehydrogenase via LC-MS analysis and protein identification. The identified heat shock protein was characterized by oxidation of the methionine residues. Okay, so just now we have seen that the metabolic efficiency of normal diet fat rats was higher. This is because they need more food, okay, in order to obtain a gram of body weight. And the palm oil based HFD, we use palm oil based, only contain traces amount of cholesterol. That's why it didn't cause high elevation of serum cholesterol in the high fat diet rats. And finally, the visceral adipose tissue of both feeding diet indicated that the calorie gain by HFD was stored as the visceral adipose tissue. Although it did not cause significant gain, but on the long run, this is expected that accumulation of VAT by HFD can lead to obesity. Okay, in terms of the protein data, HFD was found to cause changes in the protein expression in brain tissues. However, the 2D jet electrophoresis have shown that the number of spots are relatively similar. But in terms of expression, there are at least two proteins that were found to be expressed at least twofold in the brain tissue of HFD rats compared to the normal diet rats. One of the protein is the HSC70, which come from the family of HSP70 chaperone, a protein family that protects the proteome from stress. So, overexpression of this protein was also reported in Parkinson's disease. The other protein, which is the pyruvate dehydrogenase, the differential expression of this protein suggests an alteration of glucose metabolism and possibly the oxidative stress in the brain by HFD. So, as the conclusion, fat content of a diet is an important determinant of energy intake. The upregulation of two chronic stress-related proteins, HSC70 and pyruvate dehydrogenase, indicate that brain was experiencing oxidative stress and alteration of glucose metabolism as the result of HFD at its early stage of consumption. And the data also indicate a short period of excessive fat intake has an impact on the brain at the molecular level much earlier than the onset of any standard clinical indication. Alright, I'm almost at the end of my presentation. So the take-home messages are the LCMS is a powerful and invaluable tool for understanding biology. It can accelerate the biotechnological and biomedical research. However, it needs to be coupled with optimal sample preparation strategy and appropriate data analysis, which is specifically the bioinformatics. Finally, I would like to acknowledge my current and previous students, my collaborators that involved with the projects that I presented today, from collaborators from USM, FELDA, and the Malaysia Rubber Board, and finally to the staff of ABRC USM. Thank you for listening to my presentation today. I hope you have learned something useful from my talk. Thanks also to the organizer for inviting me to give this talk today. And if you have any questions, do let me know. Thank you.